this is my office, which is much bigger than my office in real life, and also much neater than my office in real life. Although there is a fruit fly floating in the coffee cup, which actually does happen sometimes. Frequently, when they're transferring the fruit flies, they will get away, and where they usually wind up is in the dean's office in his coffee cup. So I try not to let that happen, but sometimes it does. So this is where I have office hours. You see my office hours for last spring are listed up there. And so I just come in, log in, the kids know I'm going to be here. So they have questions about anything, they can come at these times and chat with me about it. And I also frequently just drop in from time to time to see what's going on. So the tower is where the molecular genetics is, and also some of the fruit fly genetics, some bacterial genetics, some human genetics. All of those things are represented in the tower. This is some of the early history of how DNA was first identified as the material, as the chemical that held the genetic information about more about the chromosome and also more about one of the genes on the chromosome. These little arrows that you see here show where the gene that the chromosome tells you about is located on the chromosome. So this is just kind of an overview of the human genome. Giant models of ribosomes. And proteins are synthesized in several stages. The messenger RNA, which is a copy of the DNA information, first has to bind to a ribosome. And then transfer RNA, which is another kind of RNA, carries the amino acids and matches up with the messenger RNA. So you can get base pairs between different kinds of RNAs, just like you can get base pairs between the different strands of DNA. And that principle is used to match specific amino acids, which make up proteins, to the different coding units on the messenger RNA. So each messenger RNA can bind to a ribosome to tell that ribosome how to make a particular kind of protein. Okay, well, what bioinformatics is about is a kind of combination of computer science and genetics. There are a huge number of databases available now that have protein sequences and DNA sequences and RNA sequences and genomic sequences and sequences for ribosomal RNAs and all kinds of things. And that information can be used for a variety of purposes. One of them is to identify organisms that you may not even be able to see. This boardwalk gives some of the genome, some of the many genomes that have been sequenced for different eukaryotes. And most of the genomes are some of the reference organisms that were first sequenced, like horses, cows, dogs, possums. Some of them are organisms that have been more recently sequenced, like this Komodo dragon here. The Komodo genome was sequenced a year or so ago. And if you click on these, you get information about the genome. This is where I usually start my classes. I figure that while the kids are getting used to Second Life, it's simpler if they all just sit. And this table expands. I think I won't ask you to try taxing its limits, but you probably could all sit here. Because every time somebody sits down, well, you've seen these tables before. Anytime you sit down, it puts a chair, another chair on that side. So it's very handy for conferences or for meeting with a lot of people all at the same time. Cool. Okay, so we're going to go look at the greenhouse. And fortunately, because it's Second Life, we can walk right through this back wall here and go into the greenhouse. So the greenhouse contains some simple Mendelian experiments. For example, the monohybrid cross, which just involves one pair of genes, is right here. And all of the objects, most of the objects will give you some kind of informational note card if you click on them. And in this part of the greenhouse, some of them will also give you a spreadsheet to put the data on. So what the dihybrid garden does is to illustrate the inheritance when you're dealing with more than one pair of genes. So we've got two pairs of genes represented here, either tall or short, and either red or white. So the two parents are in one of these dishes. The first generation progeny are all the same, and they're in this second dish that's rotating above the garden. And then the garden itself produces the progeny of the hybrids. Now, theoretically, you get a ratio of 9 to 3 to 3 to 1, 9 for the two dominants, 1 for the two recessives, and then 3 for the other two possible combinations of progeny. And again, you get a different set of progeny every time you click on the garden, and that goes into the chat record, and you can paste it into the spreadsheet for analysis. Sure, everybody's down here. So there's a bunch of different experiments that can be done with cats, but I think the most interesting one is this one here, which represents the sex-linked gene for orange. So that gene for orange is on the X chromosome. And so it's expressed differently in males and females. So the Cell platform has, I think it's 10 different kiosks. Yeah, I think it's 10. That have different experiments related to cell biology. So this is what used to be on Cell Island. And in addition to a number of experiments that range from study of macromolecules, experiments on enzyme activity, different kinds of cell and tissue types. There's the part that does cell division or mitosis. And my favorite part is what you can see if you click on this big red arrow here, and that is the giant model of a cell. You can actually go into the cell if you just click on the plasma membrane. It will take you into the cell. And then if you click on the nucleus, you can go into the nucleus. And then you can fly around in there. To get out of the cell, you have to do one of two things. You either have to find the exocytotic vesicle, which is over here. So you can click on this exocytotic vesicle to get out of the cell. But I actually bring them in, and I have a set of activities for them to do, assignments that are related to those, and then quizzes that are related to the assignments. But all of the information is in here.